Yeah, my name is Maxim, so I'm one of the bioinformaticians at AlphaFold in the PDBE team. And yeah, so I just want to, I think everyone's pretty familiar with AlphaFold by now, and I think I wanted to kind of focus on what can we do now that we have all this data. And I'm going to show you some of the features that we're working on as part of the database, and hopefully support some of the use cases that you may or may not be doing. Okay, I'm going to cover for the first bit about AlphaFold, but Arne conveniently covered some of it, so maybe save me a few minutes here and there. But really, I want to kind of dive into a lot about how to, as I like to phrase, navigate the structural landscape. And some of the things we're doing, so we're integrating this tool called Foldseek, which I'll tell you more in a bit, and kind of show you what you can do now with integrating these, this new functionality. Okay. Yeah, so the protein folding problem. So, you know, it's a 50-year-old problem, and as you all know, for those of you who are actually in wet lab, you know, it takes months, maybe even years. It's a real challenge to experimentally resolve structure. And so I guess some of the kind of two principles that have been, you know, occupied the structure sphere is that, you know, since structure can determine, sequence can determine the structure, as Anfinson's dogma stated, it could, in theory, as a kind of computational problem, be used to predict a structure. And then I quite like also as well the Leventhal's paradox, in the sense that, you know, if you were to go through every possible confirmation that you can get from a single amino acid sequence, it's going to take an eternity. So it's a paradox in itself because it takes you know, less than a moment to fold a structure. So that posed as an interesting challenge for the community for the last 50 years. And I think this is a really important figure to kind of remind ourselves as well. You know, the UniProt database has around 250 million protein sequences. And in comparison, the PDBE has just over 200,000 experimental structures, of which 60 are unique proteins. So you can see this gap is massive between the two. And obviously, using predicted structure models was kind of is the solution to kind of close this gap. Okay, so AlphaFold came along, and as Arne has said, so it's developed by Google DeepMind, and it was you know, shown an exceptional level of accuracy in the CASP14 back in 2020, and this was kind of a real kind of showstopper in the sense that, right, you know, now we can predict with very high accuracy these predicted models. I was going to go through this, but I think I won't spend too much time on this. So I think I'll skip over this for now. But I kind of want to focus more on what you can do downstream with the models that we get from our AlphaFold database resource. So I thought I'll just highlight again, as a reminder, some of the strengths and limitations of AlphaFold, the things it can and can't do. The great benefit is it's using amino acid sequence, you can predict a structure model. And what's great about AlphaFold now, let's change the shift in focus in the community, is that it's really a great way as like a starting point to start exploring testable hypotheses, as you know, Casper has shown you with AlphaFold what he's doing. And so there's, you know, many more use cases where you can start exploring ideas very quickly. As you know, with AlphaFold models, they often use templates from the PDBE as a kind of, as a template, if the MSA is quite shallow. So this is kind of another, what I was meant to say, it's starting model for X-ray crystallography and cryo-EM. But yeah, so the templates as well. And most importantly, AlphaFold produces 3 outputs. One of them, obviously, are the atomic coordinates of the model that you're interested in. And there are 2 confidence metrics that you should use always together to assess the quality of your model. I thought I'd cover some of the limitations of AlphaFold, the things it can and can't do. It's a very extensive list, but I'll cover some of them. So it only accepts 20 standard amino acids. It can predict 5 models per run as the kind of default parameter. But often they're generally very similar, these models. So in other words, it is unlikely to predict conformational variability from a single run. It cannot predict assembly, so that's why we have AlphaFold multimer. And we discourage you from predicting point mutations with AlphaFold because it wasn't designed for that. You also have, it was also not designed to predict the binding of ligand molecules or any post-translational modifications. And obviously, we heard a little bit about transmembrane proteins. It's not aware of the membrane plane either. So if it doesn't look right, the model, it's probably because it isn't right in terms of where the protein starts folding inside the membrane plane. And as you've heard, if the protein you're interested in is an orphan protein, the performance will go down. Obviously, there's fewer evolutionarily homologous sequences. So as a result, MSA is shallow, and that can suffer, the quality suffers as a result. So these are just kind of some of the limitations that I wanted to highlight. Okay, so the metrics. For those of you who don't know already, there is the first PLDDT, so it's a per residue accuracy. And so what PLDDT does really is it just assesses the quality of each residue in this space. So we've color-coded the model confidence based on these 4 colors. So I'm just thinking. OK, sorry, I thought I had a pointer. So we color-code residues in 4 categories, so blue being confident and red, yellow being kind of poorly predicted. So it's not very confident. And then the next equally important confidence metric is the PAE. So what the PAE is predicted aligned error, and so that is for each data point there is a value for every residue pair in the structure. And in doing so, it really measures the confidence of the relative position for any given amino acid for your given model. And by looking at and to analyze the data for the PAE, you can use this. It's visualized as a matrix. And so as you can see this, I thought this is a great video to kind of highlight. So what's happening here is we're selecting domains along the diagonal to kind of highlight how they are in structure, because this is interactive, and you can do this on our pages. But if you want to understand whether the actual relative position of any 2 given domains are confident, you would have to select the region that's shared between these 2 domains. And as you can see, in a second, right now, I'm selecting the region between these 2 domains. And since it's white, so white being not confident, green being confident, I wouldn't trust the relative position of these 2 domains. And so this is how you would analyze your model. OK, so I think in the more interesting parts of this talk. So it's all great now that we have all these hundreds of millions of structure. But the question is, how do we now navigate? We have this treasure trove of data. So what do we do? So to give a bit of background, it's kind of important to remind ourselves, is that for over a decade, sequence-based search methods have always been dominant because we've had more sequences than structures naturally. And so when you want to infer function from a particular protein that you're not familiar with, or it's understudied, let's say, often you would use, you'd build a multiple sequence alignment of domains that has a structure you probably know the function of, and then you would build a sequence profile, and then with that sequence profile you'd search and apply the entire proteomes, whatever you're interested in, and that way you can start, labeling certain protein coding genes for what function they may have. So that's always, one of the main ways to identify the function of your protein. Now, because we had so many hundreds of sequences, the methods have always been optimized for speed. And so, comparison to structure-based methods, they're very accurate. There's a few of them like DALI and TM-Align, but they weren't initially designed to scale, gracefully because we didn't have such large databases for structures. So we had this kind of, let's say, bottleneck in that sense. Now that we have this data, what do we do? Well, there is this fantastic tool called Foldseek, if you haven't heard of it yet. This tool, Foldseek, it was developed by the Steineger Group, based in South Korea. And quite simply, it enables you very fast and sensitive comparisons for large structure sets. And it has the level of accuracy and also the speed that we need. And quite simply, all you need to know about how this tool works is it essentially translates your 3D structure of interest, your query that you want to search with, into how they've defined a 3D interactions alphabet. And essentially, pipe that into their own sequence-based search tool, which is MM62. It's also developed by them. And because sequence-based tools are optimized for speed, it's fantastic, and it kind of and it's fast for that particular reason. So for those of you who don't know Foldseek, I thought maybe it would be nice to kind of show you why it's such an incredible tool for the community today. So I thought I'd focus on this one use case. I found this paper. So what they did with Foldseek was they were interested in studying bacterial protein toxins. And so there are an incredible amount of bacterial genomes out there that we don't know in the environment. And so often there are these gene islands which have a combination of toxin, antitoxin, and various other proteins which are involved with virulence or abortive phage infection. So in essence, how it communicates and interacts and attacks its neighboring environment with other prokaryotic cells or eukaryotic cells. So as you heard from Casper about those toxins, enterotoxins, right? So in a similar manner, so what they did with Foldseek was they said, right, let's identify these hypothetical proteins in genomes that are understudied. So specifically, they looked at the Thaura phenelacetica. Sorry, I probably butchered the pronunciation of that. And so what they did was they took these hypothetical proteins, and because we had models of them in the AlphaFold DB, they took these models and they searched with Foldseek against AlphaFold DB and the PDBE. And what they discovered, you can see the figure in yellow are actually known toxin and antitoxin proteins that have been characterized experimentally. Whereas the ones in blue are these hypothetical proteins in these predicted gene islands to be involved with the toxin and antitoxin relationship. And so I think if you just spend like a moment just to think, if you can do this with one gene island, imagine if you scale up to hundreds of genomes. And so hopefully this kind of gives you a little taster of the broader implications of what you can do with Foldseek. And so this here, they built actually a database here. You can read about it. So that's like on many hundreds of proteins that they looked at. And so yes, so it's a great tool. So we are actively collaborating with the Steininger group. And really the sole purpose of our collaboration is to enhance the depth and utility of the database. Okay, so the prediction pages. We will go through from kind of top to bottom what is already there and some of the things that we're developing now. You've probably seen this already. So this is the main homepage for AlphaFold DB. And so here you would search with your protein of interest or uniprot accession that you want to explore. And this is kind of one particular entry page that you would look at. So obviously you can see some metadata from uniprot about the protein name, the gene, and its description, and whether there are any experimentally determined structures for this particular uniprot accession. And then, of course, you have the 3D MolStar viewer to look at your predicted model and the PAE plot. Okay, so as I've shown you before this example where you can look at hundreds of proteins, right? But I think before we started looking at Foldseek, essentially it was just a landing, it was just an entry page where you can assess your model. But that's it. If you wanted to do something else with it, you'd have to download it and do your own analysis. So what we wanted to do is, because these structures are kind of naked, apart from the metrics, they're kind of naked, and you need to add information to it. Without any biological context, you really can't understand these proteins. So we were hoping that with Foldseek, the easiest thing you can do is just compare it with homologous proteins that are better studied to understand, well, this protein of interest that you're looking at, what else can I learn about it? So for the rest of the talk, I'm going to kind of look at varying levels of granularity, let's say. So I'm going to look at very similar proteins, an arbitrary measure, to kind of moderately similar and distantly similar. So I'm going to focus on homologous proteins. So when I say homologous proteins, in essence, I am trying to find a group of proteins that share something in common. And in this case, it's sequence and structure. So for those of you who are familiar, we kind of, in collaboration with the Steininger group, used the two-step clustering workflow. So this is where they clustered the entire AFDB. And we display this information in two separate ways. And I'm going to walk you through first the method and how we display the data. OK, so let's focus on the first part. So what the Steininger group did is they took 214 million proteins from the database, and they applied the MM62 tool. So in other words, they're clustering purely on sequence. There's no structures involved yet. And so the clustering criteria that they used is as long as it's a minimum of 50% sequence identity and 90% sequence overlap. And in doing so, you create now 52.3 million clusters. And so how do we display this information? So if you haven't seen already our entry pages, at the bottom, you will see there's now a new table. So it's called Structure Similarity Cluster. And so this data that I explained to you in the first clustering step, where we cluster only in sequence, so that's displayed as this table. And then you can see it's this tab, and it's called AFDB50 MM62. And then I'm just going to also explain the second part, and then I'll walk you through bit by bit. So then from each cluster, what's done next is we take a single protein from each cluster, which has the highest average PLDDT. And then now we used Foldseek to cluster. And so here, you start clustering based on 90% structural overlap and a threshold of less than 0.01 e-value. And so then we're left with 18.8 million clusters. And so now this data is visualized in this other tab called AFDB Foldseek. So now we're viewing homologous proteins on these two clustering steps, one on sequence and one on structure. So let me kind of walk you through what you can do with this table. So, as I showed you, you can navigate between these two tabs to look at different levels of homology. So every row that you see in the table is a single entry. And you can see, obviously, the AFDB accession, the description of it, the species associated with it, and the sequence length and average PLDDT. And what you can do in terms of functionality with this table is you can, you can sort the members of this cluster by sequence length or by average PLDDT. And additionally, if you want to filter your species, you can use the taxonomic filter on the top right to kind of filter your table so you're looking at proteins only in a specific organism. Right. It's all great that we have this table there, but probably the question is, what can I do? What can I use it for? And what's the point of this table? So I thought I'd highlight some use cases of the things you can do. And maybe it might benefit your work if you haven't done already. So one of the things you can do, which is from an evolutionary perspective, is really to explore in this structure space. I think the key message is how do you navigate the structure landscape? Is for your given protein of interest, across these homologous proteins, where is it expressed across the tree of life? Is it only bound in plants? Or maybe it's prevalent across the tree of life? Who knows? So by doing this, you get to understand a really good feel for this particular protein fold. Where does it sit in this structure space? Another, and I think the most powerful use case, I think I've kind of hinted on it, is to make educated guesses about the function of unknown proteins. So if you're looking at a protein from whatever study and there's barely any information, I highly recommend you check out the table, because very quickly you can find another protein which has certain criteria that matches it, and actually says, you know what, based on the function of this other protein, it's likely going to have the same function. So if you look at this example I've highlighted here, the protein in pink, I was kind of, I found it looking around, and there was absolutely no information on it. It's an unknown protein, and you can see the name of the protein is very uninformative. So what I did was I looked at the cluster table, and I found human, a homologous protein from human and from bacteria. And you can already see, based on the name, that it's possibly a kinase. So very quickly, I learned about my protein of interest. Another very interesting thing, the use case with the table, is in gain some structural insights about the differences or similarities. So I know it's a bit hard to look, but there's on the left side of this structure superposition, there is a missing chunk from the protein from Drosophila, the pink one. And because it's a kinase, there's actually a cavity where its active site is. But that chunk is missing and the cavity is much bigger. So potentially, maybe this function, the protein might be similar to these other proteins, but it might have lost its function. I don't know. So hopefully by exploring the cluster tables, it's going to open up more questions about your protein of interest. And the reason why I was able to identify this missing chunk was because I just sorted by the sequence length. So you'll be surprised how many proteins there are which are missing maybe 100, 200 amino acids. And it's examples like this where they just kind of disappear. And so it's interesting to investigate that. And then on a more technical level, if you're not too bound by your protein of interest, you can just sort by PLDDT and find a better predicted structure model for whatever it is that you'd like to do. And just to kind of bring the message home, so I thought I'd walk you through a random protein that I found. So you can see there is absolutely very little information about this protein, and the name is not very useful. So let's say but at least the PLDDT and PAE is great. So at least that's the good thing that's going for it. So what I did was I explored the cluster table, and you can see there's about 5,000 entries that are homologous with this protein. So what I did was I thought I'd save myself time and start searching for species that have, as part of the reference proteome, so any model organisms. And long story short, I identified one protein from yeast. And as you can see, it's got the badge that it's reviewed. It's from a reference proteome. And conveniently, when you read the biological function, it's a delta fatty acid desaturase, whatever that means. I'm not an expert in this particular protein. But the point is, very quickly, I just learned about my protein, which was just before bare, and there was no information in it. So this is just something I wanted to highlight. OK, but let's say you looked at this cluster table. Both on the sequence level and the structure level, and you still can't learn about your information. You think, right, is this a dead end? Well, no need to worry. So one of the newest features that we're now currently working on, and we're hopefully going to release soon, is we're going to let you, as the user, search with Foldseek for remote homology of distantly related proteins. So this is just like a preview. I know it looks identical, this table, but this is a separate table unrelated to the previous one. So what you'd be able to do is on your entry page, you'd say, submit a job, and you'll be able to search with Foldseek. And it's an e-value that's also relaxed, the threshold, so you can start identifying slightly more distant proteins, related proteins. And you'll be able to do as well two things. By searching, you'll be able to search against AFDB, obviously. But then a very important part as well is the PDBE. By superposing with experimentally determined structures, there's a lot of value in interpreting your model. So if you look at the table, it's mostly the same. It's a very similar functionality to the other table I showed you. But it's got additional sorting capabilities. So as this kind of metrics from Foldseek, you can sort by sequence identity and also the e-value to kind of score the hit. And I thought I'd show you as well. So if you do a drop-down list, you can actually see the pairwise alignment of the two and the structure that it matches with. OK, so I throw this out there just to kind of emphasize my point. So this is pairwise alignment of these two sequences, both AlphaFold models. And you've got the conservation. It's a Svelable conservation score. So I thought I'd ask you the question, are these evolutionarily related? And after looking at it for a while, you're probably realizing you cannot find a single column where the amino acids are identical. And just to give you an idea, it's 5.6 sequence identity, which is, if you looked at this, you'd say, right, they are obviously no way they are related. But I'm sorry to disappoint. It's actually this particular alignment. So what you see here is actually this particular domain. I searched with Foldseek, right? And I identified this particular domain. So what I did was I took this particular, yet again, another uncharacterized protein. And what I did is I searched on Interpro to understand if it had any sequence-based annotations. It was practically bare. There was no information. So by searching with Foldseek, I was able to actually identify a particular domain in my uncharacterized protein against this other protein from chicken. So it's reviewed, and there is actually a functional sequence-based annotation for this particular domain. And I gained this from another resource, Interpro, and it's actually a DNA single-strand annealing protein. So also to emphasize my point, I went to the very bottom of the Foldseek list. So this is the lowest e-value you can get from this search. And yet still, I was able to gain some information. Of course, I'm not saying these are definitely identical, these domains, and they have the same preserved function. You would need to investigate further. But the point is, I'm hoping to show you that I can, with these new features, we can equip you to start answering more interesting questions about your protein and start exploring really where your protein sits in this structure space, since there's hundreds of millions of these structures. And other use cases you can do. So again, when sequence homology is low, you can identify proteins. Potentially, you can also uncover analogous proteins, so by convergent evolution, so if you really relax the threshold, but that should be explored with caution. And as I think I hinted already, the ability to understand the diversity and function of protein structures makes this a really powerful tool to understand where your protein sits in the structure space. And as you can see, there are these two other domains that don't align in this structure. So I've just learned about this other protein that shares this domain. So there's another. It matches with this other multi-domain protein that has some other function. So I've learned a lot by just doing one search. Okay, I'm not a fan of this slide. For the last 20 minutes, I've been telling you how amazing structures are, but I think it's important to kind of bring ourselves back down to earth. So some of the limitations of structure-based search, and I noticed this with Foldseek myself, it's less about the tool and more about how you use it in terms of the limitations. So if you're looking at a protein that is poorly predicted or has, let's say, low PLDDT in general, or in fact, you have a very well-predicted domain, but there's tons of insertions in it, Foldseek will struggle with it. And this is based on my experience using it. So if for whatever reason you're searching for a protein that has very little information and you hit a dead end, it's possibly not a dead end yet. You probably have to investigate further, maybe additionally look into isolating the domain. Another limitation is, again, I keep reminding to always use the PAE. So if you have, let's say, an active or inactive protein which has a very large difference in RMSD with itself, maybe potentially that might affect the outcome of the results you get from Foldseek, because you might look for only active or inactive. So I haven't personally experienced this. I don't know. I think this is just something common sense in terms of how Foldseek works. And also, if you have two domains and you haven't looked at the PAE, and the PAE says, actually these two domains because the model shows that they're interacting, right? And you look at the PAE and it says, no, I'm not confident that these two are, the relative position of these two I'm not confident of, then probably the outcome of Foldseek might not be the answer that you're looking for. So you probably should look at them as individual entities, these domains. So these are just some of the limitations, but they're very easy to fix. Okay, and so really, I just wanted to highlight what's kind of why I came here today is just a sobering reminder that there's only approximately 200,000 PDV entries in experimentally determined structures, and now we have 900 million predicted structures such as AlphaFold EBI and ESM Metagenomic Atlas, and in solving the protein folding problem, created a new challenge, which was how do you make sense of this massive data set, this treasure trove of structure models. So hopefully I've highlighted to you that, in fact, by clustering, grouping proteins based on sequence and structural properties, you can start identifying relationships between similar proteins and understanding how, across evolution, where it's prevalent. And finally, with the new live Foldseek search, hopefully you'll be able to explore the function and diversity for a given protein structure that you're interested in, even when sequence identity is poor, as I showed you in the example. And before I close off, I thought I'd distract you with some pretty pictures. So essentially at AlphaFold DB, we're trying to continue empowering our users with new functionality and abilities to understand your protein very quickly just from the entry page alone. And so this is only as useful as it is to you. So if you have any particular use cases, or you see something that perhaps you think we should consider developing a particular feature, please either come talk to me today, or we've got the help desk. So just drop in a request and we'll try to get back to you. And on that note, I will leave it there. Thank you.